Hi, everyone. My name is Alex Kristoff. I'm a grad student here at UC Berkeley in Jill Banfield's lab. And what I'm going to talk to you about today is genome sequencing of SARS-CoV-2 in wastewater or sewage from right here in California. So at this point, I think we're all pretty familiar with the COVID-19 pandemic, which is caused by the SARS-CoV-2 virus. This is an RNA virus. The genome is about 30 kilobase pairs of RNA. It's spread throughout the world at an alarming rate. And one tool that scientists use to track the spread of SARS-CoV-2 is genome sequencing. And so how this is typically done is a patient comes into the clinic who is sick and who tests positive, and a sample is taken from usually the nose, and the sample is sequenced for a viral genome. And then we obtain viral genomes from individual patients, and we place these viral genomes in a phylogenetic tree and are able to make inferences with what's called genomic epidemiology, such as if two viruses are perfectly identical, they are probably part of the same outbreak cluster or very closely related outbreak clusters. In this way, we can sort of start to infer routes of transmission for the virus as it spreads throughout the world. But interestingly, something else you may not have heard of is that SARS-CoV-2 is also detected in sewage. And so we've seen a large number of preprints and papers at this point from around the world where different groups have been able to use qPCR to detect SARS-CoV-2 RNA quite reliably in wastewater, so from various cities and regions around the world. And the question that we had was, well, while those papers were on detection and in some cases quantification of viral load, what we wanted to understand is, well, could we do this sort of genomic epidemiology we do with whole genome sequences on the virus that's found in sewage, the same way or in a similar way to how we do genomic epidemiology on virus from individual patients. So we came up with this idea, as I know many other people did across the world, kind of throughout the summer too, in the late spring. And since April, we've been collecting in collaboration with several wastewater treatment plants in the Bay Area, what are known as 24-hour composite samples. So this is done by a machine called an auto sampler that siphons off a little bit of wastewater slowly over 24 hours. So you get an even sample of a region, an area, a building, whatever basically the pipe you're sampling services. And so especially in the East Bay, but also from San Francisco, South Bay, and Marin County, we've been collecting 24-hour composite samples on a weekly basis and sometimes more frequently since late April. You can learn more about the project that we have here at Berkeley, actually at our website now, covidweb.org. As I'll show you later, this project is led and directed by Professor Carrie Nelson, who's a wastewater expert here at UC Berkeley. And it's a really diverse team full of scientists, molecular biologists, and environmental engineers who have worked out a lot of the kinks to detection and sample processing. And we're talking about those in a minute, of these samples, which are pretty unique and required a lot of not out of the box experimentation. When we think about what these samples physically are, here we have a picture of what a wastewater sample will look like. It's not perhaps as solid as you might imagine. It's mostly liquid with lots of bits floating in it. And what our detection told us was via qPCR with a standard curve, was that on average in about a liter of this wastewater from the Bay Area throughout the summer, we were detecting, around 10 to the 5th genome copies of SARS-CoV-2. So that means in a mil, we were detecting only around 280 genome copies. So really to achieve robust detection, and especially as we're going to see robust genome sequencing, we're going to need to extract RNA from substantially more than 1 mil. In fact, so in general, as we look at some of our protocols that we've developed here, and that have been developed at other places here, we're usually extracting from somewhere between 40 to 100 mils of wastewater at a time. And we kind of worked with three different methods and two broad categories of methods for extraction. The first is what's known as ultrafiltration. This is where you use these columns and filters to physically enrich for viral particles via centrifugation. So where viral particles accumulate above the filter, and you basically are concentrating several mils down to just less than a mil, maybe 200 microliters, that then you'll purify RNA from. Then we had two other, and so a couple of notes about this method is, first is that it is going to try and enrich for viral particles. Free RNA, free DNA will pass generally through the filter. We try and lyse bacterial cells before we get to this point. So free bacterial RNA will also pass through the filter. We try and get rid of large cellular matter with a larger filter that viruses should pass through beforehand. So that we can think of that as a viral enriched fraction that we're sequencing. Our other two methods, however, are not going to be viral enriched. They're going to be basically all the RNA in the sample. A lot of that's going to be bacterial, some of that's going to be human, and sometimes weirder other RNAs as well. And so in these cases, what we're going to be doing, the first one is just simply with an RNA column. And what we're going to do is after a 5-micron filtration to get rid of large particles that might clog the filter, we're simply going to suck using vacuum filtration the entire sample through an Zymo 3P actually RNA extraction column. And we've tested many different columns. And I'll get to that in a second. We found that this one worked especially well. The other thing that we can do is we can add some milk silica to a sample in a Falcon tube and let that absorb the RNA, and then centrifuge it down to kind of create an RNA pellet, an RNA silica pellet that then we can resuspend. So our most successful protocol was really this column protocol using several Zymo columns. So we've since published this protocol because it's really the only one out there like it at the time online in protocols.io, and there'll soon be a publication describing how it performs to other methods. You can go check out this protocol here. It was really developed, pioneered by graduate student Oscar Whitney as part of our group. And really what you can do is you can have a large vacuum manifold like here in this picture, and you can put many columns on that and simultaneously suck through wastewater through many columns. Then there's a wash step or two, and then you elute the RNA directly from the column. And we found that this method really seems to recapitulate more total quantity of SARS-CoV-2 RNA from samples than some of these other methods. But then when we have the RNA, the question is, of course, well, we want to sequence it. And so the majority of the sequencing we're going to be looking at today is using an approach that uses a probe capture targeted method. And this was developed by Illumina, and we've had collaborators at Illumina who have helped us apply it more specifically. And it's called the Illumina Respiratory Virus Oligo Panel. And it's going to be several beads with oligos that match the entire SARS-CoV-2 genome, actually, as well as other respiratory viruses. And it will bind to cDNA to the viral cDNA specifically in a library to create a new library enriched for that specific cDNA. The full list of viruses targeted by this panel is here. Most important to us are SARS-CoV-2 and occasionally other coronaviruses. So basically, we'll be enriching for any cDNA of any of these respiratory viruses, RNA that happens to be in our original samples. For the most part, respiratory viruses don't get shed in fecal matter. It's a rather unique property of coronaviruses and of SARS-CoV-2 and SARS-CoV-1 that you see viral nucleic acids really shed in fecal matter and then therefore detectable in wastewater. And what we found was when we compared the capture-based approaches to unenriched RNA, so when we just simply did a raw metatranscriptome where we sequenced all the RNA without doing a viral enrichment, is we saw very little SARS-CoV-2. You can't even see it here in these graphs. There were some significant hits, but it was a handful, a dozen. But when we did the oligo enrichment in some samples, we would sometimes see quite a significant amount of SARS-CoV-2. We know that this is specific and also sensitive. We don't see any false positives, for example, in negative controls. And as you see, we'll be getting really high quality genomes that really wouldn't be possible from an informatics error here. And when we then look across a broader range of samples that we have here, so we have samples from Oakland and Berkeley and Marin County, what we see is in some samples, we can see a significant proportion of reads will often be SARS-CoV-2, sometimes up as much as 14%. For the most part, though, it's going to be lower. And it's interesting, there's a lot of sample-to-sample variability that doesn't necessarily relate entirely to just how much there was in the sample. With qPCR, we know that a lot of times the qPCR results say that the viral load is quite consistent, but you can have very different amount of sequencing results, and it can depend probably on the other contaminants in the sample and how the library preparation went, I suppose. And so what we're going to do to really quantify the amount of SARS-CoV-2 in these samples is we're going to map viral reads to the viral genome and filter pretty stringently, make sure they have perfect mapping scores and perfect identity to the genome, or near perfect identity, as we'll see in a second. And the other thing we see is when we then take our samples and we ask, well, how much of the SARS-CoV-2 genome did we sequence? So that's breadth of coverage. So the sample is up there at 100%, means we sequenced 100% of the SARS-CoV-2 genome. And we can compare that to what we know from qPCR, which is the approximate number of viral genomes that went into that sequencing reaction. And what we found was that only in the samples where we had at least 1,000 viral genomes going into the sequencing reaction, so that would be these samples here up top, were we able to really obtain a high quality complete genome for SARS-CoV-2. For when there was when there were only 100 viral genomes, sometimes we could even get as much as 75% of the genome, but most of the time we were then looking at lower numbers. The other thing that we can see here is that all three of our extraction methods did recapitulate complete viral genomes at least once or twice. Similar to patient sequencing, our recovery of high quality genomes and our rate of recovery is not perfect. Only some of our samples do we get a high quality genome for. Probably a little bit harder than it is for patient sequencing, but every patient sample that's sequenced also doesn't cover a high quality genome. When we look at then what the breadth of coverage looks like for these genomes, we can take one example, which was this one, and we can see the number of reads, so the coverage across the genome, and it's rather consistent, which is good. We're seeing the entire genome, and we're seeing several dozen to sometimes up to 100 reads at any given position. And the interesting thing, the thing that really attracted us to the key bioinformatics challenge of wastewater detection is the fact that if you think about where these viral sequences are coming from, it's not coming from one person, it's coming from all the people being who are infected in the area being serviced. And what we know is that all the people in these areas don't have the exact same strain of SARS-CoV-2. We know from patient sequencing in the areas that it's very common for people in the same area here in the Bay Area and here in California to have different strains of the virus acquired from different parts of the country and different places abroad eventually. And so what this means is that we're going to call SNVs, or single nucleotide variants, in our viral genome. And it's different from traditional SNP calling, where we're not trying to identify places where every read is different from the reference. We're going to also identify places where some reads are different from others. What happened when some reads were different from others? What it means is that some proportion of the virus in that wastewater sample, that even sample of the population, had a certain genotype at a position, and some proportion had a different genotype. To do this, so this is what we might call metagenomic single nucleotide variant calling. We recently developed a program to do this in this really exact context, and we were really kind of intrigued and excited to apply it to SARS-CoV-2 and wastewater. As we can see from other works on what's known about SARS-CoV-2 in the San Francisco Bay Area, is that there are many different strains. We're seeing strain lineages that originated likely in Washington State that have arrived. We're seeing lineages that came from New York and eventually from Europe and Italy. And since then, as I think we're aware, travel across the globe has kind of completely mixed the amount of transmission that you can see in SARS-CoV-2. So we were expecting that we would be able to also detect many strains in the wastewater. And that was the exact question. And in particular, we were really interested in asking, well, can we detect the known SARS-CoV-2 variants in our mixture of wastewater data? So we might, for one sample, like this sample from July in Marin County, we might be able to make a figure like this, where we're looking at across the viral genome, and each point is a variant allele at that position in the genome, and the height of the point from 0 to 100% is the number of reads in the wastewater sample that had that allele. And what we can see is that the ones that were high, say 100% of the reads, were also often alleles that had been detected in California patients. This is good, and it's what you'd expect. It means that these are likely abundant viral genotypes in the region, and we're seeing them both in wastewater and in patient sequencing. You can see then there are several alleles that aren't 100% of the viral reads in wastewater, but are some fraction. So some of the virus was in our sample was different from some of the other virus in our sample. And we're also seeing many of these variant positions being detected in California, United States also. And we can look at a similar graph across several more of our samples too, and see pretty similar patterns where we're successfully recapitulating many of the known variants in California. And we can start to quantify this effect. And we can do it in two different ways here. And the first thing we can see here is that if we look at how frequent a variant was in our samples, we can see that it was almost certainly observed in California patients. And then second most, there were several SNPs that were sometimes high frequency, maybe around 30%, that had been observed in the United States, but not yet in California. So other states, we've seen these variants, but we haven't seen them yet in California. And very few and rather low abundance SNPs that haven't been detected yet in patients in California or the United States that we're seeing in the wastewater. Similarly, then on the right, we can see the more samples we observed a variant allele in, the more likely it was to have also been observed by the folks doing clinical patient sequencing. But what we really want to know is, well, could that have happened by chance? Could it just so happen be that the math could work out that, well, we've randomly seen some SNPs and they've randomly seen some SNPs. So of course we would see some of the ones that they have seen, right? There's some probability of that happening by chance. And so we can compute this likelihood, and we can plot the negative log of the adjusted p-value of that overlap on the y-axis here. And we can do that. Actually, we can look at that comparison, the SNPs that we saw in the wastewater, to the SNPs seen in patient sequencing from a certain area for every single area. And we can say, well, what was the likelihood of overlap for each area? And when we do that for each area reporting patient sequencing from around the globe, we can see that the place where we overlapped with most in the SNPs present was Alameda County, which is, of course, the East Bay County that our wastewater samples are derived from. And they were also very similar to other samples, other patient samples from California and Northern California, indicating that we are really recapitulating the same geographic genetic signal that the patient sequencing efforts out there and locally are also finding. So with this, we can start to characterize some of the variant alleles that we're talking about. And here we can kind of group them in different boxes. And the first box you see at the bottom of this figure is the ones that I would call like the viral strains, viral genotypes that are very common in California. We know they're common because, as you can see in the blue dots, they're detected in many California patients. And we also see these in all of our wastewater samples. Great. Then right above that, we can see this interesting collection of alleles that had only recently been detected in California, only later in the summer. We're actually midway through the summer at this point. And likewise, we only saw them in our later time points in the wastewater. So this is indicating that this was more recent transmission to California that happened during the summer, maybe during the second wave of COVID. And correspondingly, we only saw these SNPs in July in our wastewater samples. Above that are rare alleles, sometimes seen in California patients, sometimes seen in the wastewater. And most intriguingly, perhaps, is the rows above that where I've listed out the exact alleles. And these are variant alleles that have been seen in other states throughout the summer and the spring, but have not yet been seen in California. But we did see them in our most recent date of wastewater sequencing, which in this data set was in July. And this is intriguing because it hints at a recent transmission event that we identified in wastewater, but hasn't been identified yet in California patient sequencing. And so that's what we see here. These SNPs are likely real because they've been observed repeatedly in other states like Washington State and Michigan State, but haven't yet been seen clinically in California, but are coming up in our wastewater data. And so this leads us to some really important conclusions, I think. The first was kind of the validation that we were seeing. We were seeing that if we look at our variant alleles in wastewater, our genomes that we're getting of SARS-CoV-2, it's really matching up with what's known from patient sequencing in the Bay Area, which is great because, and really depends on the fact that the Bay Area has enormous and very impressive patient sequencing efforts led by Chan Zuckerberg Biohub and the Innovative Genomics Institute, among others. And so it's great that with wastewater sequencing in a single sample, without relying on anyone coming to the clinic, right, or that the delay involved there, we were able to see the same sorts of genetic genotypes that they've identified in patient sequencing in single samples. We also saw that this was likely highly accurate because the chance of this overlap happening by chance or random errors is simply too low to have happened by chance. And finally, the most intriguing thing we saw at the very end there was that it looks like we have identified a transmission event of some viral genotypes to the local California area before we've seen that evidence of that transmission in genomic data collected in patient clinics. And so that's really, I think, the long-term goals and big picture prospects for wastewater-based genomic epidemiology. Can we detect, not only can we detect virus, not only can we identify how much, say with qPCR, but can we understand what are the strains in an area, where have they come from, and is it one or many viral genotypes? And I think we were able to see hints that we were able to start doing that sort of thing in this data set. So thank you. I'd like to acknowledge the enormous and amazing team that we have here at Berkeley. The COVID web, and you can go to covid-web.org to learn more. Project is really led by Kara Nelson. Joe Banfield, my principal investigator, has really advised us on the sequencing analyses here. Rose Cantor and Matthew Ulm are postdocs at Berkeley and Stanford who have played key roles here, as well as so many members of the rest of our team, including some collaborators at Illumina who helped develop this Illumina respiratory virus oligo panel and helped us debug it on our complex samples. Okay, thank you.
Hello, I'll be talking to you today about our efforts at the Los Angeles County Sanitation Districts to test for SARS-CoV-2 in wastewater. And so I'll begin telling you a little bit about the sanitation districts. The district treats wastewater for over 5.5 million people in the greater Los Angeles area. We operate 11 wastewater treatment plants, which are shown here, the blue dots. 10 of those 11 plants produce water which is beneficially reused in various Southern California communities for things like irrigation and groundwater recharge. So why did we decide to start analyzing for SARS-CoV-2 in wastewater? First, I'll start by saying that sanitation districts has been analyzing for enteric virus concentrations in wastewater since 1979, and we have extensive experience in development and implementation of methods for viral detection in water matrices. Back in late February, when it became clear that the virus was likely to be found in wastewater, we began discussions on what we as an organization could and probably should be doing to help with the pandemic. It was really narrowed down to three main areas of focus. One was the safety of our recycled water. Districts provide over 90 million gallons a day of recycled water, which the public could and possibly does come in contact with. So we had a responsibility to ensure that this water was safe. Second, we as an organization also have a responsibility to ensure that our employees that work in and around wastewater were not at an increased risk of contracting the COVID-19 disease. And at the time, there was very little data on the amount of SARS-CoV-2 virus in wastewater, and even little or even less data on whether or not it was infectious. And finally, we did recognize that there was potential for us to provide some wastewater surveillance data to help with our local public health departments and to get a better picture of the extent of the pandemic in our local communities. And so, knowing the urgency of the situation coupled with both an organizational as well as a public health need, we initiated a project to test for the virus in wastewater matrices. And so the project started off, had 4 primary goals that are shown here. First, we wanted to develop analytical methods to detect and quantify SARS-CoV-2 in wastewater. We then wanted to use those methods to test our final effluents at each of our treatment plants for the virus. Go on to determine if the SARS-CoV-2 is present in raw wastewater, and if so, what proportion is infectious. And then finally, we wanted to determine if the SARS-CoV-2 signal in wastewater can and does reflect the state of the pandemic within the community. So my talk today is going to focus primarily on the analytical methods for quantifying SARS-CoV-2 in wastewater, but I'll touch a little bit on their correlations with some of the pandemic metrics in the end. If there are questions on some of these other areas, though, I'll be glad to answer those at the conclusion of the talk. Okay, I'll begin by telling you a little bit about viral testing in wastewater, a little bit of background here. First of all, we recognize that the data is only going to be as good as the methods that are used to obtain it. So before we can attempt to try to use this data for surveillance or to evaluate risks that are associated with wastewater, we need detection methods that are sensitive and reliable. And so with viral analysis in wastewater, they generally consist of 2 steps, a concentration step followed by then detection or quantification. Most human viruses are in low concentrations in wastewater, and therefore they need to be concentrated to bring the signal within a range that can be detected. There's a couple ways to do this. One is to concentrate the viruses within the wastewater itself. And then second, if you're using a PCR or a genetic-based assay, you can look at concentrating the nucleic acids after they've been extracted. The overall goal of the concentration step is really to increase the sensitivity of the detection, which is dependent on both the recovery efficiency as well as the overall concentration factor. Now, all concentration methods are going to have some loss associated with them. And so really the aim is to maximize your concentration factor while minimizing this loss. And then following concentration, the viruses need to be detected or quantified. There's 2 ways you can do this. It's either by cell culture or by genetic methods such as PCR. Due to the biosafety concerns with the SARS-CoV-2 virus, detection now is almost exclusively done through genetic methods using RT-qPCR or digital RT-qPCR. There are multiple SARS-CoV-2 PCR assays that have been described, but in general, the nucleocapsid N1, N2 tests developed by the Center for Disease Control are the most prevalent in the United States. Okay, so the method that we use is a direct extraction method. The workflow is kind of outlined here. And as I go through it, I'll highlight some of the areas that we believe are important to obtaining sensitive and reliable data. Starting with the sample collection, we collect 24-hour flow-weighted influent wastewater. I will note that we have also collected grab samples at peak flow, and we don't see much difference between the 24-hour composite and the peak flow grab samples. Upon arrival at the lab, the sample is placed into an RNA preservative, stored at -80 until the RNA is extracted. The use of a preservative allows us some flexibility when processing, as it prevents RNA degradation from RNases or freeze-thaw effects. So it enables us to retain the signal strength while also reducing variation. Currently, we're using Zymo's RNA DNA Shield. There are other products out there that can perform or at least achieve similar outcomes. Another critical component to any kind of quantification approach really involves assessing the recovery of the process. This is especially important when you're looking at wastewater, where you can have things in the matrix that can interfere. So without some metric of recovery, you're not going to know if the differences you see in the concentrations of the virus are due to processing issues or changes in the viral RNA in the wastewater itself. And so there are several types of these sample processing controls that have been described, ranging from other coronaviruses to bacteriophage. But because our process is based on direct RNA extraction, we use an in vitro transcribed RNA that's not normally found, the sequence isn't found in nature as our sample processing control. This gets put into each sample prior to extraction, goes through the complete extraction process, and we can quantify it at the end. The advantage here with the in vitro transcribed RNA is the reagent doesn't require cell culture to produce like some of the other viruses. It can also be easily and accurately quantified. We have also evaluated OC43 coronavirus as a sample processing control, and in our hands, it performs similar to the in vitro transcribed RNA with this method. Now, as I stated a little bit before, wastewater by nature is a complex and dynamic matrix, which can lead to increased variation, especially when using molecular methods. Replicate analysis can help to mitigate the variability associated with wastewater. This is the approach that we take. With each of our samples, they're extracted a minimum of 3 times, along with a mock extraction control to control for cross-contamination. And moving on to the RNA extraction step, the purpose here is to extract and concentrate RNA with minimal loss, and also to reduce or eliminate inhibition. We found that including the solids portion of the wastewater increases signal strength, which isn't too surprising. The SARS-CoV-2 virus has been shown to associate primarily with solids in the wastewater. There are several commercial kits available. We currently prefer Zymo's quick RNA fecal soil micro kit. For us, it kind of checks all the boxes on what we're trying to do with this step. In particular, it allows us to extract RNA from both solids and aqueous fractions. There's also 2 inhibitor removal or impurity removal steps. The kit includes also a concentrating column that allows elution volumes in as little as 5 microliters. So a lot of advantages that we like, with the Zymo kit. And finally, final part of this process is the detection and quantification, which in our lab we're currently using RT-qPCR with the N1 and N2 primer probe sets. What I'd like to highlight here is that SARS-CoV-2 is an RNA virus, and so you're going to get the most accurate quantification by using RNA-based standards. And so for our assays, we in vitro transcribe RNA standards for both N1 and N2, and that's what the quantification is based on. All right, I'll briefly go through some of the kind of critical parameters that we've determined, for this method. Starting with the method detection limit, we've determined that the detection limit in residential wastewater is approximately 5 × 10⁵ copies per milliliter. This was done by spiking decreasing amounts of heat inactivated SARS-CoV-2 into residential wastewater that was negative for any native SARS signal. We generally see recoveries of above 70% for this method, and this is based on that in vitro transcribed RNA sample processing control. Also, the CT values from the RT-qPCR of that processing control generally are within half a cycle of one another in the samples and the mock extractions, and this kind of highlights the consistency of the method overall. We do see on occasion some inhibition. Usually, this is observed in wastewater samples that are composed of both industrial as well as residential inputs. And we found that diluting the sample 1 to 2 or 1 to 4 usually mitigates the inhibition. All right, so to further evaluate our approach, we did split sample analyses of wastewater influent with 4 other laboratories. The other laboratories all concentrated viruses within the wastewater prior to extraction, but used slightly different methods. Showing the results of those experiments here. Lab 1, sorry about that. Laboratory 1 would fail to recover any virus signal in any of the samples tested. Their internal processing control recovery was less than 0.1%, so lost most of the virus to their process. The data from labs 2 through 4 showed that the concentrations were between 1.2 and 2.8 logs lower than the direct extraction method. They were also more likely to have results that were negative or below detection. And we also observed less variability with the direct extraction method compared to the other wastewater concentration methods. And so kind of thought this provided some favorable metrics to the direct extraction methodology. And so I'll sort of summarize some of the advantages and disadvantages to using this type of method for detecting SARS in the wastewater. Starting with simplifying workflow, there's less processing involved, and so this kind of increases throughput, allows us to analyze more samples at once. There's minimal loss during the process, which kind of makes sense. Silica-based RNA extraction purification chemistry has been well characterized over the years. It has been shown to have a high recovery with relatively low variability. The method also minimizes variability as a whole, as we saw in the last slide, and really the main disadvantage to this method is that you're limited to processing smaller volumes. That's going to equate to smaller concentration factors, which are usually on the order of 10 to 100-fold. All right, so using the direct extraction method, we have collected longitudinal data of the SARS-CoV-2 RNA concentrations in influent coming into 2 of our largest plants in Los Angeles. Shown by the red lines are the weekly average of SARS-CoV-2 concentrations in the wastewater. The black dots and black lines represent the 7-day average of new COVID-19 cases in Los Angeles County. For the wastewater samples, the concentrations do represent the average of the N1 and N2 values. We do this because we do get slightly different values for N1 and N2, and at this time we don't know if one is more valid than the other, and so we are averaging the 2 values. As you can see, both the wastewater data and the number of new cases show similar trends, indicating or at least suggesting that the wastewater data will correlate with the extent of SARS-CoV-2 that's found within the community. Note that on occasion we do see outliers. This is kind of exemplified by the point on the plot to the left. We actually think that this has more to do with the sample collection than the analytical process. As far as the sample to the left, this particular data point was analyzed on multiple occasions with each of the data points showing the elevated concentration. Additionally, there are some other wastewater parameters that we look at on these samples that were also kind of out of the norm for this particular sample. So I would like to point out that while we can perform the analyses and identify trends of SARS-CoV-2 concentrations in wastewater, we don't claim to be epidemiologists, and so we think that any further interpretation of the data really needs to be done in collaboration with public health officials. All right, I'll finish up by describing some of the challenges that we've encountered with the SARS-CoV-2 testing. The primary, the most substantial of these were supply chain issues. This ranged anywhere from PPE to RT-qPCR master mix to RNA extraction kits. We've seen some improvement of late, but with the current spikes occurring in Europe and the Midwest, we're anticipating some additional shortages in the future. The second issue is sensitivity. The method is limited by small volumes, and while the values that we've obtained have been within the detectable range of the assay, the levels that we've seen as of late are nearing the detection limit. Despite that LA County still has a positive test rate of between 2 and 3%. So in the future, additional sensitivity may be necessary, and we're currently investigating other approaches to achieve this. On occasion, we do see inhibition. It is rather sporadic and usually occurs in mixed wastewaters composed of both industrial and residential inputs. And that diluting the samples has been pretty effective in abrogating the inhibition for the most part. Finally, the last issue I'll talk about is interpretation of the data. Wastewater surveillance is still a relatively new approach in the United States. And as I mentioned last slide, we can test the wastewater and come up with values of SARS-CoV-2 in the wastewater and determine how those trends are occurring over over a longitudinal basis. But really, further interpretation should be done in collaboration with federal, state, local public health departments. They have more expertise in the area, and so that's kind of the way that we've gone about this in talks with our local public health department. And also along these lines, we're participating in the CDC's National Wastewater Surveillance Initiative, in which they're aiming to help local health departments better understand the extent of COVID-19 infections in their communities through wastewater surveillance. And so that project for us is going to start up relatively soon. And so I'll end here and thank some of the people in my lab that helped produce the data that I've presented here today. And we can take some questions at the conclusion of the talks.
Hi, everyone. I'm Raul Gonzalez. I'm an environmental scientist at Hampton Roads Sanitation District in Southeast Virginia. And our group has been doing HRSD's COVID-19 wastewater surveillance for the last few months. This talk is really focused on our methods and our workflow and how we optimize assays as they come into the lab, specifically for community level monitoring on pooled wastewater samples. We took our first samples and analyzed those samples in early March. And the reason we were really able to get up and going was because we actually already had an environmental surveillance program where we were monitoring for antibiotic resistance and various reference pathogens. So it was a fairly easy transition. And a little bit later, I'll talk about what it took to actually optimize and validate those assays here at HRSD. But first, the map you're looking at is the Hampton Roads region. Again, it's in Southeast Virginia. And most of our work focuses around the raw wastewater of our 9 major facilities that you can see there. And in this map, what you're looking at are the actual catchments for those wastewater facilities. If you're listening to this, you probably already know that there's 2 kind of main methods being used to quantify SARS-CoV-2 in wastewater. The first is a culture-based method. It requires a really high BSL level, and this determines infectivity. But I think most of us are using PCR-based methods, like RT-qPCR and digital PCR. And these PCR-based methods don't differentiate between viable and non-viable viruses, but they do tell you whether the genetic material or the RNA of the virus is present. Now, kind of honing in on those PCR-based methods, I think the majority of the workflows can be broken down into these 4 steps. So once you collect your sample, there's sample concentration, essentially where you're concentrating large volumes and concentrating down those viruses. Then nucleic acid extraction, reverse transcription, since SARS-CoV-2 is an RNA virus, and PCR quantification. And there's many iterations within each one of those steps. For instance, for sample concentration, some of the popular concentration methods are PEG, electronegative filtration, ultracentrifugation, and various others. For nucleic acid extraction, there's you can do RNA extraction only, or RNA and DNA co-extraction, and within those there's many options. And then even for reverse transcription, you have the option of 1-step or 2-step. And as I mentioned before, for PCR quantification, you can do qPCR or digital PCR. Now here at HRSD, this is our workflow. This is our current workflow. We've tried many iterations for each one of these steps, but for us, we're really trying to maximize throughput and minimize costs. So this is kind of what we've come up with. For concentration, we really like electronegative filtration, or that's essentially using HA filters that are negatively charged, and then we put a charge on the sample and run that through. For nucleic acid extraction, we do total nucleic acid extraction. We actually have this automated. The reason we do total nucleic acid extraction is because on a given sample, we can run a whole suite of assays, everything from SARS-CoV-2 to adenovirus. So we definitely so we've realized that we like both DNA and RNA. For RT, we like one step. Again, this is for throughput and cost. And I think I mentioned this earlier. Our preferred PCR method is digital PCR. In our experience, we find digital PCR to be more sensitive in environmental matrices and specifically in wastewater. Now, to get into the weeds a little bit more, just to kind of dial in on this, as I mentioned before, this isn't the first iteration of this workflow, even for this COVID-19 wastewater surveillance, but our current assays that we use are N1 and N2 multiplex, and then we use bovine coronavirus as a spike in every single sample, and so of course we quantify that, and then we use pepper model virus as kind of like a housekeeping gene for wastewater. We don't really show a lot of analysis normalizing for pepper or using pepper model virus to normalize our data because we do most of our analysis on load, on either instantaneous load or total load, but we have it there just in case. In terms of volumes, this is a question that we get asked a lot. How much volume do we process? It's always it's a balancing act between detection limits. So the more volume you assay, the lower your detection limit, but you also increase matrix inhibition and matrix effect. So for us, our sweet spot is assaying 100 mils of sample. This gets us a detection limit of about 1.9, 3.2, and 2.3 copies per reaction for the N1, N2, and N3 tests or assays. Or if you like to see those in copies per 100 mil or copies per 100 mil, it's about 47, 81, and 58 for those three assays. Now on tough samples, on samples that have a lot of matrix inhibition, we assay somewhere between 25 and 50 mils. To get into a little more detail on the processing, we don't let samples filter, and this is an in-house policy. We don't let samples filter for more than 20 minutes. If 100 mil of wastewater doesn't go through in those 20 minutes, we just reassess. So we either do 250 mil samples or 425s, and then just adjust the total assay or the total volume assayed on the back end by extracting the multiple filters and combining, or some iteration of that. When we're filtering, we also always process a filter blank or two, and we do this on a batch basis. So if we're running our weekly wastewater epidemiology samples and samples for a different project, we'll run potentially just one filter blank for all of those. When those filters get to the lab to start the molecular workflow, every single batch of samples that are extracted also get a negative extraction control or a blank filter, and that kind of carries over onto the PCR plate also. So when we run a PCR plate, we run 2 no template controls that bookend our samples. We include that that negative extraction control, our samples in duplicate, the filtration bank, and positive controls. So digital PCR doesn't need a standard curve, but we do need positive. So we use a standard that we buy, as well as generally we use a wastewater standard as well. Our recovery. So currently we're using bovine coronavirus as our main recovery spike in, and with that we get approximately about 6% total recovery for our workflow. We have tested other ones like BRSV and hepatitis G as well, and we get pretty similar recoveries for our workflows. Now, what I just showed in the previous slide is kind of like our standard workflow for our weekly samples. But every once in a while, we do supplemental samples to get more information out of our workflows. For instance, one of our more popular routine additions is running straight sample, so unconcentrated samples. We do this on about a monthly basis. So our total nucleic acid extraction instrument allows us to extract up to 2 mils of samples. So we process 2 mils, which does bump up our detection limit from approximately 50 copies per 100 mil for 100 mil samples. That it bumps up the detection limit to about 10 to the third copies per 100 mil. But we are benefiting in better recovery. As you can see, our bovine coronavirus recovery of just a 2 mil sample is about 60% on average, and for BRSV is about 75%. But you can gain a lot of information from unconcentrated samples. For instance, you can calculate what I call indigenous concentration recoveries, or the recovery of only the HA filters. For instance, I put on the slide the indigenous N1 concentration recovery for the HA filter on this particular week was 11%. And you can also calculate this for BCOV and various other assays as well, which I've also put. BCOV on this week was about 4%, and pepper mottle virus was about 8%. So that's the recovery of only the HA filters. We also get asked a lot about optimization. So back in February, when we were optimizing the CDC diagnostic panel to wastewater, we kind of have like a checklist of things we do in the lab. So prior to even ordering the primer and probe set, we always check like the assay design. So we check whether the melting temperature, the GC content, the product size kind of meet the design specs for our instrument, which for us is the Bio-Rad QX200. And so you can see here on the upper left of the slide, I did this for N1. So I know these assays are designed for qPCR, and they're not ideal for our instrument, but we're able to make them work just the same. But these melting temperatures here seem a little out of range from what Bio-Rad recommends for their instrument. The GC content looks good, but the reverse primer and the probe were flagged for high-end self-complementarity, which means it's predicted that some primer dimers might form with these. So I always check these before ordering. I think the N2 assay, if I remember correctly, was a little closer to the design specs for assays or for preferred assays on our instrument. After this, once we get the primers and probes and our standard in stock, or once we get into our lab, we always run an annealing temperature thermal gradient, and then figure out the theoretical detection limit. So for the annealing temperature, that's what you're seeing at the bottom left. This is a figure of us optimizing the distance between our baseline droplets and our positive droplets. Apologize for everybody who's using qPCR. These are figures for our droplet digital PCR. So once we've optimized the annealing temperature, we then go ahead and do theoretical detection limit estimation. So our instrument calculates detection limit a different way than I think most people are used to, but I still like to use this this method that we borrowed from the qPCR world, where we take a standard or wastewater that has the target of interest and do serial dilutions at a high number of replicates till you don't see detections anymore. And so I call that our theoretical detection limit. And then something that I feel like doesn't get done enough is determining the assay specificity and sensitivity. So the specificities can be done actually very easily in silico. If for the CDC diagnostic panel, for their paper that describes all of it, they did a really good job of describing how to get the specificity, which I believe for the N1 and N2 assay we're near 100%. And then sensitivity is a little more difficult. What I'm showing at the bottom is our false negative rate for our assays. This was determined. We did something a little different for this. What I did is, so I know what zip codes fall within our catchments, and I also know where to get health clinical data from the health department by zip codes. So essentially, I know what period in time for each one of our treatment plants there were infected people in the catchment, and then I just tabulated and aggregated the number of non-detects for each assay during that time frame, and that's how I generate these false negative rates. But I'm not saying that this is what everybody needs to do. This is just like the first kind of basic steps that our lab does that we think is worth doing. And these can all be done for qPCR as well. Probably should be done for qPCR as well. And I'm coming up to my time here, so I thought I'd just end with like a last thought. I know that there's a lot of, especially for those of you who probably aren't used to running qPCR or RT-qPCR, that it might seem like there's a lot of controls and a lot of standards and kind of redundant tests in there, but I find it's really important to run all of them and on every sample. This is the bovine coronavirus recovery for the last 2 weeks in October for our weekly sampling, and I've always thought wastewater to be very consistent in terms of its reference pathogen concentration, its fecal indicator concentration, but I find it's not consistent in terms of matrix effects and potential inhibitors to your PCR. So if you look at our bovine coronavirus recovery for the second to last week in October, we actually had pretty high recoveries. We actually had these for a couple of weeks in a row. And then all of a sudden, the last week in October, there was a sharp decline in it, and it's kind of stayed this way. And there's a lot of potential reasons for that, and I have my own guesses. But there's also, besides week-to-week changes, there's actually changes on plant-to-plant wastewater. So we have facilities that that consistently have really low recoveries, like this facility here, which I know is due to saltwater intrusion and also a high percentage of industry in that catchment. So it's really important to like run all of these so that you really do understand what your results mean and helps put the numbers into context. With that, I'll go ahead and stop and take any questions.
So Raul, I just got asked a great question by Maria Burgos-Garay, and she said, do you need to change the annealing temp from qPCR to ddPCR for the N1, N2 when you guys, or if you did try between both? You mentioned that you stuck with ddPCR. Was there a transition? How did that
In general, every time we onboarded Assay, we always do an annealing temperature and adjust accordingly. But we did actually happen to run qPCR side by side with our digital PCR early on. We did this as like a multi-lab study with Dr. Noble's lab at the University of North Carolina, and it was actually similar for both. It was, I think it's 55 degrees. But across the board, we always do an annealing temperature check just to see if the assay even works, and then go from there.
Yeah.
All right, I have a question being asked here. What form of bovine coronavirus is being added as a spike in? Is it the virus itself?
Oh, I guess that's for us. It's actually a vaccine, and we actually have a paper where I think it links you to the one that we use. We get it from a veterinary supply company. It's in water research. I bet if you Google Southeast Virginia COVID wastewater, it'll come up, and it should link you to the actual product that we use. I don't know. Are you guys using bovine, Ryan and Alex? Are you guys using bovine coronavirus as well?
We've looked at OC43 and bovine coronavirus. We kind of think the OC43 is a little bit more performs a little bit more similar to the SARS-CoV-2. But for us, because we don't really concentrate on the front end, we're kind of sticking with the in vitro transcribed RNA, a little bit more accurate on the quantification, I think, on our end. But when we've done at least comparisons with the OC43 and the in vitro transcribed RNA, we see pretty similar recoveries.
Yeah, similarly, the group at Berkeley is also using BCOV. It's the vaccine, like probably the same stuff you have. It's a little bit. We've seen varying recoveries with it that kind of haven't led us to validate it completely as a spike in, but other people could tell you more about that than me, I think.
Yeah, Ryan. Actually, maybe it's a good opportunity to note that we also tried OC43, and we're considering it as a spike in, but we have background OC43 in our wastewater at about 10 to the third per liter. So we decided to scrap it after that. I think we tested it for a few weeks and saw it.
Yeah, we see a little bit. It's kind of site specific, but we end up spiking in like 2 logs higher than what the background would be. But that's a good point, Raul.
Just to sort of segue from there, because we seem to be getting a lot of questions towards the beginning of the workflow, specifically with the auto sampler that I think all 3 of you guys incorporated into your workflows. Have you guys by any chance incorporated anything to account for viral degradation that may be present at that step, if there is some? I know groups are slightly worried that if they are collecting over 24 hours, water that's collected toward the beginning of a time 0 might degrade, and that's all dependent if it's a viral capsid or free RNA at that point.
I can talk a little bit on this. I think Raul's also done some work with this. So early on, we looked at doing both grab samples and composite, we didn't see much of a difference. We have done a small kind of time course to look at the stability of the viral RNA signal in wastewater, where we just pulled a sample, let it, incubate it at room temperature for 48 hours. Didn't see much of a change. There is a caveat there in that our samples were inhibited, and so we're going back now and going to perform a little bit more detailed test on that. So it seems that the virus, at least in that one small study, that viral signal did not degrade too much over the 48 hours. And then our direct comparison with the 24-hour composites and the grab samples didn't show any statistically relevant difference.
Yeah, I'll just add to oh, sorry. Oh, was someone going to say something else?
Go ahead.
I'll just add to Ryan's comment in that for about 6 months from March, starting in March, for about 6 months on the sites that we had 24-hour flow-weighted composite samples, we actually also concurrently collected a grab during the collection of the 24-hour composite, and just kind of overall didn't really see a difference for us. The collection time was around peak flow, which I always recommend if you're going to do a grab, that's when you should collect it. So we didn't really see a difference there between the composite numbers and the grabs. And that went for all of the assays that we were looking for. And our travel time, so that might be relevant to interpret that statement. Our travel times from our plant, I think the max travel time is like a day. And then of course, smaller than that.
And I have kind of a follow-up question on that one, which is, do you keep your samples on ice while you are collecting?
We do, yes.
I think it can vary from auto sampler to auto sampler.
Yeah, we have, so we have two different types of auto samplers that we use. One is actually refrigerated, so it keeps the composite at 4 degree. But then we have other ones that we do ice regularly while they're collecting.
So Ryan, question for you, because you showed some interesting data what other labs were sort of doing as opposed to you guys doing direct concentration. Do you by any chance know what methodologies, whether that be a PEG precipitation or something they were doing towards the front end that caused sort of this discrepancies between your limits of detection versus theirs?
Yeah, and I should, as I was listening to my talk, there's a kind of an important typo in one of the units. The detection limit is actually 5 × 10⁵ copies per liter, not milliliter. But as far as that, so we've looked at doing some PEG precipitation early on, and we did see kind of a large loss in the viral signal on the precipitation end. We've done some other work on the final effluent, and where we're concentrating, hundreds of liters or so, and we also see that this virus just doesn't seem to like to precipitate very well, and so we think that's part of where that loss is occurring. One of the I guess kind of advantages of this direct extraction is that we don't have to do any of that pre-processing, so there's not a lot of steps in there where we're losing virus, and kind of adds to the higher recoveries that we see. But I think there's we participated in a WRF study, and I think Raul's group also, and Kara Nilsson's group, we're all in there, and the conclusions from that were that if you take into account recovery, all of these methods are kind of valid. They each have their advantages and disadvantages that you can search through and kind of pick out the best method for your own particular use.
Great. And I have a question here from Jen Vanderklen. What is the exact, especially for Ryan, what is the volume you're using for direct extraction?
Yeah, so we've modified a Zymos kit a little bit. One, as I kind of mentioned, we mixed the sample with the RNA shield stabilization or kind of preservative, And so we also use that as the lysis buffer. So we don't add additional lysis buffer, but the volumes that we're collecting are 250 microliters. And so the other reagents in that kit are scaled up accordingly.
All right. And a question from Anda, specifically addressed to Raul Gonzalez. Our filtration pretreatment of adjusting the pH to 3.5 and 25 mils magnesium chloride both necessary? Is adjusting pH to 3.5 sufficient? And a question to Alex. Well, actually, I will go on to the question to Alex in a moment. Let's see if Raul can get to that one first.
I think you just we tried various iterations, and I know others have as well. I don't think it's necessary. I actually just heard from a lot of I've heard from several other people that actually don't do any adjustment to the wastewater, and that they find that they get higher concentrations from no adjustment at all. I would just try all the iterations of it in your own lab and see which one works better. For us, acidifying and adding magnesium chloride seems to give us the highest concentration and the best recovery. So that's what we went, but I know others don't.
All right, and this is the first question that we're going to get to this one. Somehow I don't think it'll be the last. And this is the dreaded clogged filter question. So this one is directed mostly at Alex, but I think everybody here is going to have something to say on this. So with the sodium chloride lice samples, how easily do they tend to clog filters? Both the PVDF variety filters membranes and the Zymo column. So, and this is especially when you're dealing with a sample that is more solid content.
Yeah, I mean, from experience, they can clog for sure. We found that the specific columns being used could vary enormously in their capacity to clog. Really, the ones that seem to clog the least were those Zymo 3Ps. We compared a couple other types of Zymo and actually some other columns from other companies too. The 3Ps didn't seem to clog as often, but still depending, and we've also noticed that depending on where your wastewater comes from and what your sample looks like, some samples are far more prone to clogging than others. It's a difficult question, I think. You can filter on 5 micron and 0.45 micron before going through the column. We found that in cases when we have to do that, we think it can reduce recovery a bit if virus is stuck to larger particles. Yeah, that's something I think that we found that almost every different really sampling site and source of wastewater, you might have to optimize a little bit differently for possible clogging. The strength of your vacuum can make a big difference. Yeah.
Yep. Any tips from Raul or Ryan on how to best avoid clogging situations?
Yeah, we kind of find we have a variety of different types of wastewater, and I agree with what Alex is saying. It really is kind of site to site. And I think some of Raul's data that he kind of showed at the end just goes to show you that just how dynamic the wastewater can be, and you kind of have to take it on a sample by sample basis, and it reinforces why we should be using the recovery controls and things of that sort. But I think probably Alex or Raul have more experience on the filtration end of this than I do.
Yeah, we echo what you guys said. We definitely get really interesting samples. Actually, when we got LA County Sanitation District wastewater, it was night and day from our wastewater samples. I probably wouldn't have even had I known it was going to be like that, I wouldn't have said that we were going to concentrate the samples. I would have just ran them straight. But yeah, for us internally, that's why we have that 20-minute kind of filtration time. If a volume that we want to process doesn't filter in that amount of time, and we're not going to adjust our protocol, then we just adjust the volumes and get creative. And we can process about 100 mils through an HA filter. 50 goes through really easily, but if we really want to target 100 mils, we'll just split in half and then do 2 extractions and elute half the volume and collect. Or our extractor allows us to just extract 2 filters at once. So we just kind of got to get creative to get to that same volume assay. I think that's the trick.
Interesting. So essentially what I'm getting from all of you guys is that it's almost site dependent, because there's so much discrepancies between whether that be more industrial runoff, et cetera. So that sort of poses the question, if you're like Alex, who's in, you know, I know he's not processing samples directly for Berkeley, but if he was like looking at dormitory samples, they would sort of need that additional concentration step rather than like a direct extraction. Yeah, which is really fascinating.
And then jumping off of that one, so actually 2 things. Number one, we've also been doing some experimentation in-house with wastewater, and actually just like Ryan, we've been focusing a lot on seeing what we can do with the fecal soil kit, and that's actually led to kind of an evolution of that kit. We started off with that one, but obviously made some changes, and we will soon have available, I think actually in the next couple of days if it's not out already, a kit purpose designed for dealing with wastewater.
And just as a follow-up, so everyone's sort of, you know, classified their routine to process the influent. Is there a reason you guys have sort of avoided primary sludge?
Yeah, we've looked at primary sludge, and what we see is, one, just dealing with the matrix is very problematic. You know, the sludges at a lot of our plants are kind of like mud, so it's basically trying to extract RNA from something the consistency of a very thick mud. So just pipetting is an issue. You also have holding time, so that those primary sludges aren't giving you any, you know, you can't really take a 24-hour composite of a primary sludge. So, you know, exactly what that value represents, you can back calculate a little bit, know what the, you know, the kind of precipitation or flocculation rate is. But for us, we, you know, we were able to you know, we calculated roughly about a, you know, a 10-fold concentration in the signal in the primary solids, but we had to dilute it 1 to 10 to 1 to 50 to be able to get rid of the inhibition. So it kind of canceled out any kind of concentration factor advantage that we were seeing. So that's kind of primarily where we just decided to focus on the influent.
Cool. And going also back to the question of controls and recovery, I have a question from one of the attendees, actually a few attendees. What is the best method for using the pepper mild model virus to calculate your percentage recovery, since all wastewater will have that virus present?
Oh, if that was specifically geared at me, that was, so that's not why we use pepper mild model virus, but when I pair a concentrated sample and an unconcentrated sample, I can get the recovery of pepper mild model virus from the filter only. So the recoveries that we're looking at right now are like total recoveries from, you know, concentration, DNA extraction, RT, all that combined. And sometimes we just like to know like what the breakdown is of loss. So I know that we lose most of our template in like the concentration step. And so every once in a while, I just like to know, okay, just check how much am I losing for a bunch of different targets, you know, from concentration only. Alex, I know you guys are doing pepper mild model virus too, right? But as a normalization.
Yeah, I mean, so just so we're all on the same page, I guess there are 2 controls right here. One is for a coronavirus recovery control, and that's where you can use like a B-cub spike in. And the other is like a fecal loading control, which is PMMOV, which is, so we're using the endogenous PMMOV levels to understand how a sample might vary in really its proportion fecal matter, if that makes sense. And so, yeah, the folks in Kerry's lab have developed a separate qPCR assay for PMMOV, and really are taking both in longitudinal time series, and one is basically one is being normalized to PMMOV. I've seen other groups out there like Eric Alm's group that have shown that PMOV is like pretty stable over time, or at least not drastically changing over time in their watershed, or no, you know what I mean, sewershed, and so right now, yeah, we are normalizing to PMOV as an indicator for the amount of fecal matter that might have been in a sample.
And a question directed at actually very much at Alex. So the question is, when was the presentation recorded? And I'll just answer that one. Say the presentation was recorded, I believe, on Wednesday night last week. But I think more what they're getting at when they ask, have there been any updates since then, is if I remember correctly, the data being presented in your preprint was collected around the summertime. Is that right?
Mm-hmm. Yes.
And I imagine there's still some ongoing work there. Are there any new developments relative to your talk and/or the preprint that you would like to talk about?
So we've moved on for the most part due to some life goes on for people on the team, and really what Kara's team is now super focused on is on specific locations, in some cases individual institutions and buildings, and communicating really q-RT-PCR data to public health departments and to decision makers in a couple of different contexts. So what I mean is that while what I presented on was really these sequencing results, and we think it was a cool kind of proof of concept, to get that down to a few days information that would be a few days turnaround is very challenging on the sequencing side. And I think we would be a year away from that, whereas we have to communicate information to decision makers like now. So for that reason, Kara's team is really focused on communicating q-RT-PCR results in several contexts around the East Bay. So to answer the question, we really haven't done any more sequencing since then. For example, where the team has done a lot more q-RT-PCR, and so that's sort of the development we've seen here. And as you can see, q-RT-PCR is certainly not easy, and quantifying viral load is certainly not easy, and sequencing adds a whole nother level of complexity to that.
Alex, you bring up a great point there that I kind of wanted to address before. So essentially, you guys are relaying this RT-qPCR data to public health officials and decision makers. Now, how is the data being reported to them? Because I know, for instance, Ryan at LACSD was averaging the two N1 and N2 concentrations together for one, final copies per liter. Is that something that they'd encourage, or what's the sort of, if how they like the data to be reported?
Yeah, I'm not really the expert, and I think in many cases it's sort of an informal relationship. But in several cases, I think they like to see everything. It's important to look at, has your N1 gone up? For example, the N1 PMMOV ratio, you have to be a little bit careful about. Has PMMOV been the one changing, or has N1 been the one changing? We also have an ESAR-BACO assay for another region that we're looking at simultaneously. And yeah, so long story short, showing kind of several results, not just one final number, so that they can really have the context for the data, because I think it really hasn't been worked out in a formal context how to formally interpret that data. I think is very challenging. So really communicating multiple numbers and being open and honest about what all the results show at any point. And
All right. I have somebody double checking with Raul. Can you describe your direct extraction method?
Oh, it's actually the same. Well, for this work, it's the same extraction method as our concentration samples. It's an automated system that uses chemical lysis and magnetic beads. same process. It just allows for 2 mils of direct sample.
Excellent. And I have a request from Fernando Roman from UC Merced. Looks like they're working on using the geographic information systems to start mapping out some of these wastewater findings. So I'm going to put in the chat a link to the map project that they're working on. I had a quick look at it during the presentations. It's pretty cool, so I will definitely send that out. Thank you, Fernando. And I believe I am at the end of my question list that I've been sent. Michael, did anybody send you any additional questions?
No, none so far that I see. If you guys have any, quick additional questions, please feel free to send. But if not,
I'll take this silence to apologize for the yellow tinge. I don't know what's up with the camera.
I think we've hit the end of everybody's questions. So I'd like to thank everybody for attending. It's definitely been a great webinar. As I mentioned earlier, we're about to launch a wastewater specific product. And in addition to that, I guess I didn't give my own affiliation. So along with being a team member at Zymo, I'm also adjunct at UCLA. So I know about this also because I've been assisting some UCLA laboratories with trying to implement wastewater monitoring on their dormitories in the near future for when the students return to campus. So I'm a little bit on both sides of the equation here. And as I mentioned, we're about to launch an environmental monitoring product, specifically, it's a Zymo Environ Water Kit, Water RNA Kit. And it's one of our first kits that's specifically optimized for getting viral RNA out of water. And oh, I do have a new question coming in. What volume of the let's see. How much RNA recovered from 250 microliters of wastewater did Ryan assay in a single RT-qPCR reaction?
good question.
Yeah, so we have looked at the total RNA concentration. We don't know exactly how how accurate that's going to be because we're at the end of our reactions we're only left with 1 or 2 microliters left, and so we do use a dye-based kit to kind of get those concentrations, and they do vary a little bit, but usually around I think it's 3 to 10 nanograms or so.
Cool. So, and then saying what volume?
Our elution volume is 15 microliters, so 250 microliters of wastewater eluted in 15 microliters of the buffer, and so roughly about a 17-fold concentration factor.
Excellent. All right, and I believe, yep, I think that's the end of the questions that I have. So, Michael Lysick, anything additional on your end?
No additional questions being sent to me. I just want to say thank you guys for all the panel speakers and those tuning in today to watch a lot of great presentations.
Mm-hmm. As I mentioned at the beginning, this is being recorded, so I will in the next couple hours, once this is done processing, I will cut it all together, and we should be able to send out a link to everybody. We'll probably be posting this on our YouTube and potentially a few other places. So as soon as I have it cut and rendered down, I will send out links to everybody. All right, thank you. It's been a good morning.
All right.
Thanks.